Throughout history, there have been many reports of living animals being discovered encased inside of solid rock and other hard enclosures. This phenomenon has understandably been met with skepticism, because it seems impossible for an animal to survive in conditions to where they seemingly have little to no access to nutrients, water, or oxygen for as long as they've been trapped inside of these enclosures. How exactly could an organism manage to live for such a long period of time in such destitute conditions? And although these reports usually involve frogs, there are several other types of animals that have been discovered alive in similar conditions after people have broken open rocks that these animals had been encased alive inside of. Numerous studies have demonstrated that animals are endowed with biological mechanisms that can allow them to survive for very long periods of time in the most extreme of conditions. But is it really possible for an animal to survive indefinitely in such conditions? Benjamin Franklin documented the phenomenon of live entombed frogs in depth, writing at Passy, near Paris, April 6, 1782, being with Monsieur de Chaumont viewing his quarry. He mentioned to me that the workmen had found a living toad shut up in the stone. On questioning one of them, he told us they had found 4 in different cells, which had no communication, that they were very lively and active when set at liberty, that there was, in each cell, some loose, soft, yellowish earth, which appeared to be very moist. We asked if he could show us the parts of the stone that formed the cells. He said no, for they were thrown among the rest of what was dug out, and he knew not where to find them. We asked if there appeared any opening by which the animal could enter. He said no. We asked if, in the course of his business, as a laborer in the quarries, he had often met with the like. He said never before. We asked if he could show us the toads. He said he had thrown 2 of them up on a higher part of the quarry, but he knew not what became of the others. He then came up to the place where he had thrown the 2, and finding them, he took them by the foot and threw them down to us upon the ground where we stood. One of them was quite dead, and appeared very lean. The other was plump and still living. The part of the rock where they were found is at least 15 feet below its surface, and is a kind of limestone. A part of it is filled with ancient seashells and other marine substances. If these animals have remained in this confinement since the formation of the rock, they are probably some thousands of years old. Toads shut up in solid stone, which prevents their losing anything of their substance, may perhaps for that reason need no supply, and being guarded against all accidents and all the inclemencies of the air and changes of the seasons, are, it seems, subject to no diseases, and become, as it were, immortal. Even Carl Linnaeus, the renowned biologist, believed that it was possible for amphibians to live for centuries or even millennia encased inside of rocks, based on his examination of numerous reports of the phenomenon and his extensive studies of animal biology. Another renowned naturalist, named Dr. Robert Plot, who worked at the University of Oxford in England as a professor of chemistry, provided even more examples of live frogs and other animals being discovered encased inside of rocks and other solid enclosures, writing that toads were often discovered inside of stones without any perceivable rift or cleft. And Plot was privy to this phenomenon having occurred in many different places throughout England, such as in Horton, where stonemasons were breaking rocks to build a barn, and inside of one of the rocks, they discovered a toad, which was still alive when it fell out of the rock, but died soon afterwards. And Plot described another account in which a toad was discovered alive inside of a hard limestone block that had been used as a stepping stone. People who were walking along the path kept hearing a muffled croaking noise, and they couldn't figure out where the sound was coming from, but eventually someone realized that the sound was coming from inside of one of the stone blocks, and deciding to break it open, they discovered a cavity inside of the stone, and inside of the cavity there was a large toad which was about the size of a fist, and the toad, which was still alive, hopped out of the stone after it was broken open. Plot also wrote in his text, quote, "In the coal mines near Ley, There are oftentimes found hard, round, smooth flints with living toads in them, without any perceivable cleft or passage for air. Platt wondered to himself in his book, writing, "Now how these animals should come to be thus included in the middle of such entire and solid substances. How, when such imprisoned in so narrow a cell, they're supplied with the necessities of air and element? To which I answer that these animals require very little of either to support them, for the cavities that they're lodged in are generally somewhat bigger than themselves, and they have the salts of the stones, which together with the transcolation of such fine dews may very well support an animal that no way spends itself in perspiration, and is absolutely shut up from all other expenses of its juices or spirits, in swimming, traveling, generation, or otherwise. But to what period of time an animal such imprisoned and secluded from observation is not easy to conceive, much less to determine, though I believe one may venture, in general, to pronounce it a pretty long one." And Platt also described other animals, aside from frogs and toads, such as newts and lizards, which were also found alive, encased inside of rocks. There have been over 200 reports of animals being discovered alive after being removed from rocks and other solid materials in which they were fully encased for an extended period of time, far longer than their usual lifespans. In 1982, railway workers near Takuti in New Zealand were clearing away a dense layer of stone to extend a track. And 12 feet below the ground, one of the workers pointed out that after he had broken away some of the stone, he found inside a small cavity which contained a small frog, and the frog had been fully encased inside of the stone. The next day, another worker discovered a second living frog, embedded deep inside of another section of the stone that they were trimming away. In the year 1818, a geologist and natural historian named Dr. Edward D. Clark, who was giving a lecture to students at the University of Cambridge, said that he had discovered several live newts sealed inside of chalkstone that he was chiseling while he was looking for fossils at a depth of 270 feet below ground level. While he was digging, he had collected several small newts that he had found inside of the chalk, which had belonged to the supposedly extinct species Megalotridon. Clark had laid the newts out on a sheet, and he saw that 3 of the newts began moving. After a short period of time, 2 of the newts stopped moving, but the third one continued to periodically move its appendages. He brought the newt that was still moving to a nearby pond and put it inside, and once the newt was rehydrated, it was able to swim away. In the year 1773, 2 men were working at the Nybro quarry in Gotland, Sweden, and as they were quarrying large blocks of sandstone deep below ground level, they discovered a living frog inside of a small nodule in the sandstone. The frog appeared to be lethargic, but it moved, and it was visibly breathing through its nostrils, but its mouth was covered by what the quarrymen described as a yellow membrane, which is consistent with the type of mucus coating that frogs and toads are able to produce to prevent themselves from dehydrating during long-term dormancy. Both of the quarrymen were shocked to have discovered a live frog embedded inside of the sandstone that they were quarrying, and they reported their discovery to the inspector of the quarry. The inspector followed the quarrymen back to where they had discovered the frog, and the frog was still there. The inspector confirmed that the frog was alive when he saw it, but when he touched it, it barely reacted. And so he took one of the quarrymen's shovels and hit the frog with it. After he beat the frog, the inspector regretted doing so, and he took the frog and brought it to various researchers. And this frog was brought to the natural history collection of Count Carl Gustav Tessen at the Akero Castle in the 1700s. According to an 1865 edition of The Zoologist, workers excavating limestone 25 feet below ground level in Durham, England, discovered a large living toad encased inside of limestone that they had removed. One of the workers stated, quote, "The cavity in which the toad had been contained was no larger than its body, and presented the appearance of being a cast of it. The toad appeared, when first discovered, desirous to perform the process of respiration, but evidently experienced some difficulty, and the only sign of success consisted of a barking noise. On a minute examination, its mouth is found to be completely closed, and the barking noise it makes proceeds from its nostrils. The claws of its forefeet are turned inwards, and its hind ones are of extraordinary length, and unlike the present English toad. The toad, when first released, was of a pale color and not readily distinguished from the stone. But shortly after, its color grew darker until it became a fine olive brown. The workers had the toad and the limestone it was encased in brought to the Hartlepool Museum, where it was put on display. In the Waratah tunnels of Lambton, England, there were several workers mining for subbituminous coal. According to the workers, they discovered, quote, in a place without any outlet whatsoever, a live frog of a most peculiar species that had been fully encased inside of the coal until it was broken free. The frog was about 3 inches long and had unusually large eyes, but the eyes were glossed over, and the frog did not respond to the workers' gestures, indicating that it was blind. And several other living frogs and toads have been discovered encased inside of coal by workers at the Swimtillery colliery in Wales, as well as by coal miners in Maryland in the United States who had been excavating lignite. The New York Times reported on December 24th of 1906 that in Cumberland, Maryland, a coal miner named John Savage, who worked at the Enterprise mine, which had been used for mining bituminous coal and lignite, had discovered a live frog after breaking open a lump of coal, and the frog had jumped out of the coal after it was released, and the frog hopped around Savage's feet. In the 1500s, a man in Moudon, France, named Ambrose Paré, was having a conversation with workmen who were quarrying stone near his house, and Paré saw one of the workers break open a rock in which, quote, a huge toad full of life fell out of it and hopped away. In the year 1851, a man in Blois, France, broke open a large nodule of flint with a pickaxe, and inside of the flint, there was a living toad that fell out of it. The toad proceeded to crawl away, but the man picked it up, and he sent the toad, along with the flint that it was encased in, to the local Academy of Sciences to be examined, because he thought it was such an unusual find. And the Academy of Sciences performed a series of analyses on the toad, during which the toad remained alive, and the researchers confirmed that the toad had survived fully encased inside of the flint, and that the flint had formed rapidly around the toad, encasing it inside. After taking note of the various reports of live entombed animals, the Academy of Sciences in Paris conducted an experiment in 1771, in which the researchers there encased 3 toads in plaster and sealed them in a box for 3 years. When the toads were taken out of the plaster after 3 years, 2 of them were still alive. Later, in 1825, a professor of mineralogy at Oxford named Dr. William Buckland conducted his own experiment to see if toads could survive encased in stone for long periods of time, having himself been skeptical of the numerous reports. Buckland entombed 24 live toads inside of a cavity between 2 blocks of limestone that were sealed together using plaster. A year later, he broke open the block, and over half of the toads were still alive. And a naturalist named Philip Goss assessed Buckland's experiment, and he concluded that more of the toads would have survived if they had been in their torpid, hibernating state at the time that they were entombed. According to a 2009 herpetology report, thermoregulation, performance, and energetics, for amphibians, rates of water loss can be extremely high, and most species select microhabitats that minimize water loss. Such microhabitats are usually relatively cool or enclosed. These conditions, in which people have discovered living frogs and toads encased inside of rocks, are comparable to the microhabitats that these researchers are describing, which contributes to these amphibians' ability to retain their moisture and remain alive in a state of suspended animation for thousands of years. Frogs are extremely hardy creatures that can survive in a variety of harsh and otherwise unlivable conditions by becoming dormant. They're able to survive droughts and dehydration through a process called estivation, which is a prolonged torpor or dormancy, which many types of animals can enter into when there's a severe food shortage or a drought. Even in conditions to where a frog almost completely dries out, they're able to become dormant, and they can reanimate once they're rehydrated, with frogs being able to bring their bodily functions to a near halt in order to conserve their energy until their situation improves. And so, in the cases to where the frogs discovered encased inside of rock appear to be dried out and dead, they may have actually still been alive in estivation. And Benjamin Franklin actually confirmed this, saying that one of the dried toads discovered by the quarrymen had been preserved in spirits of wine, and although they had believed the toad to be dead, it actually reanimated once it was rehydrated in the spirits. And there were many other types of animals, such as turtles and some fish, that can be completely dehydrated, but they can remain alive by estivating and going dormant until their bodies are rehydrated. There are some areas where there used to be ponds and lakes in Death Valley, and when the area dried up, the fish burrowed beneath the dirt, and were able to survive by becoming dormant until it rained again. These mechanisms allow the animal population to survive, even during severe droughts. When frogs estivate, they grow additional layers of skin as a means to prevent water loss. The minerals that they're surrounded by when they're buried sometimes partially permeate their external dermal layer, which is why many of the frogs and toads that were discovered encased alive inside of rocks had been the same color as the surrounding stone, and after emerging from the rock, they shed their extra layers of skin and reverted to their original color. And many amphibian species are also able to coat themselves in a thick mucous membrane during estivation, which also prevents them from dehydrating, which is also often reported by people who discovered live frogs encased in rocks. One common excuse that skeptics use to explain away the phenomenon of live frogs being found alive inside of rocks is that the frogs have been simply hopping around in the area, and the people who thought that they saw the frogs inside of the rocks actually just saw a frog near the rock, which is a major assumption to make. But if this were the case, then the frogs that these people were seeing would not have been coated in a thick layer of mucus, because amphibians only produce this during long-term dormancy. All animals require fuel and oxygen to produce energy for their bodies and to maintain their cellular functions. The organisms that can survive for extended periods of time in suspended animation generally do so by conserving the energy stored in their bodies in the form of fat and other reserves. Eventually, those energy reserves would run out and the organism would die, but in the case of animals capable of cutaneous respiration, energy sources can be obtained from the materials surrounding the organism. Any animal that has a moist, impermeable skin is able to utilize cutaneous respiration to obtain resources. In order for frogs to absorb oxygen, electrolytes, and nutrients through their skin, there has to be a certain level of moisture present. And the types of rocks that these frogs have been discovered encased alive inside of, such as limestone, sandstone, flints, and low-grade coals, are permeable, so they can allow moisture, as well as oxygen and minerals, to seep through them. And this keeps the frogs moistened, hydrated, and provides their bodies with enough oxygen and minerals to survive in dormancy. And being submerged in water allows a frog to acquire oxygen, because frogs are able to breathe underwater, even without having gills, due to their skin's respiratory capabilities. And just to demonstrate precisely how much minerals, gases, and water are able to seep through rocks, even solid rock, such as limestone, take into consideration how stalactites form. When water passes through the ground, it reacts with carbon dioxide in the soil, producing carbonic acid. The acidic water dissolves the limestone beneath the earth, and it gathers other minerals as well, as it leaches through the rock. And once the water has traveled through the ground deep enough to reach a hollow cavity, such as a cave underground, it releases carbon dioxide and deposits calcium carbonate, and forms stalactites on the roof of the cave. And this is the same way that geodes usually form. If there's hollow cavities inside of a rock that are created by gas bubbles or dissolved matter that had been inside of the rock, then eventually this cavity gets filled with mineral-rich waters, and the minerals get deposited inside of the cavity. And if there was a living organism inside of this cavity, such as a frog, it would take these minerals leaching through the rock and absorb it into its own body, as well as the water that brings the minerals in. The types of rocks that these animals have been discovered alive inside of, namely limestone, sandstone, flint, and low-grade coals, are also themselves very high in trace minerals, and the frogs are able to absorb these minerals through their skin as well. While some compression is typically involved in the formation of these types of rocks, they can form without significant compression. Both organic and inorganic limestone varieties can form through a process called chemical precipitation, where calcium carbonate dissolved in water precipitates out and accumulates, and therefore a frog could become buried in sediment during a flood, and the high levels of calcium carbonate in the water would help to lithify the sediment that the frog is buried in, still alive, and preserve the frog inside. And likewise, sandstone typically contains calcium carbonate and iron oxide. As the sediments settle underwater, the minerals in the surrounding water crystallize and are deposited within the pore spaces between the sand grains, forming a cementing material, without the need for compression to any significant degree. Hence how a frog could find itself inside of this lithified sediment without having been crushed during the lithification process, because the process of lithification doesn't require compression. As mentioned before, frogs have also been found encased alive inside of flint too. And flint forms when silica precipitates and fills spaces in calcium carbonate, hardening into flint by cementing, with no compression or heating required during the lithification process. And in the frequent cases in which frogs have been discovered alive inside of coal, this consistently involves lower-grade coals, such as subbituminous coal and lignite. And coal forms when plant matter called peat decomposes, and both lignite and subbituminous coal require far less heat and pressure to form than standard coal, hence how these animals were able to survive inside of this lithified peat without being crushed or superheated. In fact, some lignite consists of up to 70% water, and typically still contains plant debris that's not yet formed into coal. Lower-grade coals, such as lignite, are also very low in potential toxins, such as sulfur and carbon. And so lignite, and even subbituminous coal, are essentially very dense soils, making it a rather comfortable and safe environment for a frog to hibernate inside of, until being broken free eventually by coal miners. One of the most unexpected discoveries of a live animal embedded in rock was described in the February 9, 1856 edition of the Illustrated London News. According to the report, French workmen had been excavating sediment in order to build a railway tunnel between Saint-Dizier and Nancy. The workers broke open a large mass of limestone that had been buried, and inside the rock there was a live pterosaur. After the workers broke away enough of the stone, the pterosaur fell out of it, and the workers said that the pterosaur had fluttered its wings and made a croaking sound before dying. They described the pterosaur as having around a 10-foot wingspan, and 4 legs joined by a membrane, and a long mouth full of sharp teeth, and a black leathery body that was very oily, not dissimilar to a salamander. The workers brought the pterosaur to the town of Gray in France in order for it to be examined by paleontologists. But after the pterosaur was accepted for examination, it was never seen again, and the workers were unable to receive any updates about it. Based on the workers' description of the pterosaur, it may have possessed skin capable of cutaneous respiration, explaining how the pterosaur was able to obtain resources while dormant, encased in stone. But many researchers believe that pterosaurs had a covering of fur or protofeathers based on their examination of fibers on fossilized specimens. So what is true? The reports of the workers who encountered a live species of pterosaur, or researchers' interpretations of fossilized pterosaur specimens? To answer this question, In 2007, two researchers, Thea Garden-Lingham Soliar and Alan Fiducia, had examined a species of small dinosaur called Sinosauropteryx, which was believed based on previous superficial examinations to have been covered in a layer of protofeathers. The researchers obtained the Sinosauropteryx fossil, and they analyzed it using scanning electron microscopy, and they determined that the structures resembling filaments were in fact not feathers, protofeathers, or even fur, but rather they were partially decayed collagen fibers, and they were actually located within the confines of the Sinosauropteryx's body, not on the surface of its skin, as fur or feathers would be. Lingham Soliar and Fiducia tested their suspicions that these structures resembling protofeathers on the Sinosauropteryx were actually partially, and not even fully, decayed skin by taking dolphin carcasses and burying them for a year, and then examining the partially decayed dolphin carcasses using scanning electron microscopy and chromatographic analysis. In doing so, the researchers confirmed that the partially decayed collagen fibers present in the dolphin carcasses were identical to the fibers preserved in the fossilized Sinosauropteryx, and they were also identical to the fibers previously falsely believed to be protofeathers in fossilized pterosaurs and ichthyosaurs, which the researchers also analyzed using scanning electron microscopy, and revealing that there was no evidence of feathers or fur in any of these fossilized organisms, and that the fibers seen were just the remnants of skin. Lingham Soliar wrote in his report, "These degraded fibers present in the dolphin carcasses presented almost limitless pattern permutations, in many instances showing feather-like patterns that are strikingly similar to many of those identified as protofeathers." The researchers concluded that all reported cases of protofeathers in dinosaurs, marine reptiles, and flying reptiles alike show a striking similarity to the structure and levels of organization of dermal collagen. And the type of skin that pterosaurs have, based on this study, indicates that they were capable of cutaneous respiration through their highly vascularized, furless wing membranes. Reptiles such as pterosaurs also have low levels of maintenance metabolism, allowing them to conserve more energy, while being able to absorb resources such as water, oxygen, and minerals through their skin. It would not be biologically feasible for any animal, especially a large one such as a pterosaur, to remain dormant for at least 65 million years, though, as mainstream scientists claim that the last pterosaurs thrived on Earth. Even while dormant, an organism has to carry out some biological processes, such as cell division, and long before even 1,000,000 years have passed, the telomeres in the organism's cells would have become too short, resulting in death, because there would still be cellular division, albeit at a far slower rate than when the animal was not in hibernation. However, if pterosaurs had actually existed before the worldwide flood, which was only 4,500 years ago, and this pterosaur became encased in sediment that cemented around it during the ancient cataclysm, it would be much more plausible for it to survive for only a few thousand years in suspended animation, as opposed to millions of years. The oil-like substance that the workers reported seeing on the pterosaur's skin may have been the same type of lipid and wax-based compounds that modern reptiles produce to serve as waterproofing and surfactant agents, similar to the mucus coating that amphibians produce. And this oily substance produced by the glands in the pterosaur's skin helped it to preserve itself while it was dormant. The pterosaur's large size, however, explains why it died quite soon after its euthermic arousal. Larger animals have more difficulty awakening from hibernation, with the difficulty increasing the longer the animal has been dormant. It takes significantly more energy to raise their body temperature back to normal after a period of lowered metabolic activity during hibernation, making the process of waking up much more energy intensive compared to smaller animals. Essentially, they need to burn a larger amount of stored fat to warm up their entire body. So, a large organism, such as a pterosaur, would need to consume a very large amount of food before hibernating in order to have enough energy to resume activity after an extended period of time remaining dormant. Overall, this report is plausible because there is sufficient evidence to support the possibility of a pterosaur having remained dormant since being catastrophically buried in sediment that had cemented around it until the pterosaur was eventually discovered by workers who were digging a tunnel thousands of years later. when the pterosaur had revived after being exposed, it used the last of its energy reserves, and it was unable to survive reanimation, especially considering its large size. It makes sense that the pterosaur would have been able to stay alive while dormant, with its body conserving its energy and lowering its metabolism to the bare minimum required to survive. But its arousal expended the last bit of its energy, and it promptly died after moving around. The reason why amphibians and reptiles are able to survive after being encased in rock for very long periods of time, aside from their ability to acquire resources via cutaneous respiration, is due to the fact that they're ectothermic, which means cold-blooded. Ectothermic organisms do not require energy to warm their bodies, like warm-blooded animals do. The amount of energy that endothermic, or warm-blooded, animals require to heat their bodies is the primary reason why they need so much energy in the form of food, as well as their constant need for water and oxygen to perform cellular processes. Warm-blooded animals have higher metabolic rates than cold-blooded animals, which means that their cells have to produce more ATP, which requires glucose and typically oxygen. In the process of converting ATP into energy, called hydrolysis, also requires water. And while cold-blooded animals also produce ATP, they do this at a far slower rate than warm-blooded organisms. And so, cold-blooded animals, such as amphibians, reptiles, and even insects, arthropods, and worms, require far less food, water, and oxygen than warm-blooded animals. And this is why only cold-blooded creatures are able to survive after being encased in destitute environments for very long periods of time. Warm-blooded organisms, such as mammals and birds, require a constant intake of calories to maintain their own internal temperature, which requires a lot of energy, and therefore they're able to quickly run out of energy and starve. Whereas cold-blooded creatures don't require energy to generate body heat, and therefore their energy requirements minimal, provided that they're sitting still and not moving around often. The aforementioned examples of live entombed animals involve amphibians and the pterosaur, rather than other types of animals, such as mammals and birds. If a type of animal that is not capable of cutaneous respiration is buried, they're not able to absorb sufficient nutrients or oxygen from the permeable rock around them through their skin in order to maintain their vital functions long term, without awakening and eating food. And a warm-blooded animal will not be able to survive for long in a destitute environment, even through dormancy, because warm-blooded animals' bodies require too much energy to allow for long-term dormancy, with certain warm-blooded animals only being able to hibernate for approximately a year. Whereas cold-blooded organisms, especially those capable of acquiring resources through their skin, are able to survive for very long periods of time in suspended animation, whether they be trapped in lithified sediment or any other solid enclosure. As for the pterosaur, which was found alive inside of a limestone boulder, there is scientific evidence indicating that pterosaurs are capable of cutaneous respiration, unlike most other reptiles, because of the pterosaur's very large and highly vascularized wing membranes. Although most types of animals do not have the biological capability to survive in suspended animation for thousands of years, because they're not capable of acquiring additional resources through their skin when they run low, the way that animals capable of cutaneous respiration are able to. Animals can survive in a variety of catastrophic conditions without access to the types of resources that they would need to survive when they are active. Hibernation, estivation, brumation, and torpor are all ways that animals can conserve energy and slow down in response to adverse environmental conditions, with the specifics varying depending on the environmental factors and the species of the animal. But all of these processes can simply be referred to as suspended animation. It's essentially like a sleep mode that different types of animals can stay in for longer or shorter, depending on their biological capabilities and ecological factors. Many types of animals are able to remain in biological stasis for very long periods of time, when adverse conditions prompt them to retreat without having access to resources, provided that they have sufficient lipid stores and are well hydrated. For example, the European dormouse can hibernate for up to 11 months in the wild. And the Australian eastern pygmy possum was documented to have hibernated for up to 367 days. Some species of snails, such as the African giant land snail, can hibernate for up to 3 years during unfavorable environmental conditions. And the reason why land snails can remain dormant for so much longer than mammals is because land snails are cold-blooded, so their bodies have far fewer energy requirements. And they're capable of cutaneous respiration, meaning that they're able to acquire resources through their skin while they're dormant. Bears are not technically capable of true hibernation because they don't depress their biological processes or lower their body temperature enough, but in every other way they're essentially capable of hibernation. Most types of bear are able to remain dormant for 7 months at a time without having to get up once to eat or drink. And provided that they have eaten enough beforehand, it is feasible for them to remain dormant for even longer periods of time. So in theory, most types of bears can remain dormant for even longer than 7 months, but they have no reason to do so in normal conditions. There are some other species of animal that don't typically hibernate, but they have the physiological capability to do so in specific conditions, such as extreme cold or during food shortages or droughts. This includes small birds, such as the common porwheel and the chickadee, as well as squirrels. These animals do not hibernate every year, but in the cases of extreme food scarcity or other adverse conditions, they're able to enter into a state of dormancy. There are many different bat species that do hibernate regularly during the winter, but this trend does not apply to tropical bats. However, if tropical bats are faced with food shortages or low temperatures, they also have the ability to enter into a state of dormancy by slowing down their metabolism and conserving energy for long periods of time. Long-term hibernation can also significantly prolong an animal's life far beyond their natural age range. In laboratory studies of mammalian hibernators, long-term induced states of dormancy have been found to result in nearly a 10 times higher lifespan for certain species, such as Siberian bats, ground squirrels, marmots, and mouse lemurs. Each of these species would live far shorter lives if they did not enter into regular hypometabolic states of dormancy, which preserve their cellular processes and significantly decelerate processes that contribute to aging. For example, telomere shortening is one primary contributor to aging. Telomeres are DNA sequences at the ends of chromosomes that shorten with each cell division. By significantly reducing the rate of cellular division while dormant, these animals preserve their telomere length because their cells are dividing less frequently, which helps them to live for longer than usual. Recent studies on European dormice and Jungarian hamsters show that these animals are actually capable of increasing the length of their telomeres in the active season following hibernation, which partly contributes to the increase in longevity in these animals. Exposure to adverse ecological factors, such as air pollution and ultraviolet radiation, are also believed to reduce lifespan. But when animals hibernate, they generally take shelter, such as by burrowing underground, which protects them from these potential aging factors. A frog that's been dormant inside of a rock also suppresses their metabolic processes, reducing their rate of cellular division, and are protected from environmental factors that can contribute to aging, which, like the other animals described, can prolong their life far beyond their typical age range. A 2020 study by Rasha Alatar and Kenneth B. Story explains the reason why animals in dormant states are able to live abnormally long lifespans. The authors state that a common theme amongst hibernators is their ability to greatly reduce their metabolic rate to a fraction of their normal rate and initiate cytoprotective responses that enable their survival. Metabolic rate depression is strictly regulated at different biological levels in order to enable the animal to not only survive, but to also do so by relying mainly on their limited internal fuels. At the cellular level, aging is associated with a decrease in reparative and regenerative abilities in tissues and organs. For example, when the body is under stress, it uses its physiological reserves to restore homeostasis, such as repairing damaged DNA, proteins, and damaged tissue, with greater stress requiring the use of more physiological reserves. Over time, the physiological reserves deplete, and when the aging body is faced with stress, it's unable to effectively restore homeostasis. Hibernation involves a significant decrease in metabolic rate, which is thought to slow down these cellular aging processes. Animals are also able to remain in the same positions for long periods of time in dormancy, and not experience the same types of health issues that they would face if they remained in the same position when they're not dormant. Studies have shown that while animals are hibernating or in estivation, they don't suffer from muscle atrophy. This is because, while these animals are dormant, they can utilize a mechanism in which they can reabsorb nutrients from their waste products, such as urea from urine, and this reduces the need to break down muscle in order to replenish amino acids. Due to these mechanisms, an animal is able to remain dormant, laying in the same position for years, and experience no significant damage to their muscles. Many insects, arachnids, mites, and other organisms undergo a process similar to hibernation, called diapause. Diapause is a response to adverse environmental conditions, such as changes in day length, temperature, or food abundance. During diapause, there is reduced metabolism and minimal cell division, and the organism's development halts until the environmental conditions become more favorable. During the late fall, woolly bear caterpillars seek out dark, secluded places to hibernate, and like frogs, they're able to produce a natural antifreeze in order to prevent themselves from freezing, and then they enter dormancy. When the weather warms during the spring, the woolly bear caterpillars are thawed out. In order to prolong the lifespan of night crawlers that are used for fishing bait, these worms are usually stored in fridges and packed into containers full of moist dirt that allow the worms to burrow beneath and estivate, which significantly increases their shelf life. Like frogs, these worms often cover themselves in a layer of mucus in order to prevent themselves from dehydrating while they're in estivation. Live worms also have been discovered embedded inside of solid rock deep beneath the earth. This is because they burrow into the ground, and in certain conditions, the sediment around them lithifies into rocks, which prevents their escape, and the worms remain dormant until they're released. In 2018, a research team in the Arctic discovered several roundworms, which the scientists claimed were several thousands of years old, and these roundworms were discovered encased inside of solid and lithified permafrost, approximately 40 meters, or 131 feet, below ground level. And these roundworms were still alive after being uncovered and thawed out. The roundworms were able to survive for thousands of years in these conditions by using a biological mechanism called cryptobiosis, which brought their metabolic processes and their development to a halt. Another well-known example of an organism that's able to survive an array of adverse and catastrophic conditions is the tardigrade. Tardigrades are also capable of entering into cryptobiosis, where they lose almost all of their water content and enter into dormancy. In this state, they essentially stop all of their metabolic processes, and this allows them to survive in environments with no food, water, or oxygen. And using cryptobiosis, they can also endure extreme temperatures, radiation, and even the vacuum of space. The body cavity of tardigrades is an open hemocoil that makes contact with every cell in the tardigrade's body, allowing sufficient nutrition and gas exchange with no need for circulatory or respiratory systems, much like the cutaneous respiration that amphibians are capable of. And if a tardigrade were also sealed inside of a rock for thousands of years, it would also re-emerge alive, just as frogs have been able to. Bacteria such as Bacillus and Clostridium can form protective layers called endospores in the presence of high heat, toxic chemicals, or when there is a lack of nutrients. And these spores are able to remain dormant for thousands of years before becoming active again when the conditions become more favorable. Like frogs, there are some species of lungfish, such as the Protopterus, that can enter into a state of estivation in response to drought, during which they burrow into the mud, and they can survive for several years burrowed underground. Lungfish can significantly reduce their metabolic rate, and while in estivation, they do not eat or drink. However, they cannot survive forever without access to oxygen, and this is where cutaneous respiration comes into play. Like frogs and newts, lungfish are also capable of cutaneous respiration, meaning that they can absorb oxygen and release carbon dioxide through their skin, provided that they have access to sufficient oxygen, which only needs to be a very small amount during estivation. A lungfish can survive in a barren environment without food or water for up to 3.5 years. And there have even been cases in which lungfish were found still alive inside of hardened sediment that it burrowed itself into in order to estivate during a dry season. Although primates are not typically associated with hibernation, there are actually several modern primate species that are capable of surviving in adverse conditions by becoming dormant. Dwarf lemurs can hibernate for 3 to 7 months every year. Before hibernating, dwarf lemurs eat a lot of food to store fat in their tails, which also contain water and nutrients. In the event of a disaster in which a dwarf lemur would need to seek shelter without having food available, they would be able to hibernate for long periods of time until it's safe for them to resurface, without there being any significant impact on their health. Recent findings have also indicated that Neanderthals, which are an early species of human, were also able to hibernate. Neanderthal remains found in the Cima de los Huesos cave in Atapuerca, Spain, show seasonal growth disruption, which researchers believe indicates that this early species of human may have hibernated using a similar strategy to cave bears, being able to slow down their metabolism and sleep for several months at a time, which is especially useful in the case of food shortages or adverse environmental conditions. And this would have enabled this early species of humans to hide out in the event of a catastrophe and remain dormant to conserve their energy. Based on the evidence outlined in this video, it's clear that a wide range of animals possessed a similar ability, which allowed them to preserve themselves in catastrophic conditions by hiding out and becoming dormant for long periods of time. Although some creatures clearly had more difficulty than others re-emerging from their shelters. Thank you to those who have supported Shattered History: John R. Amun, Nathaniel Lee, Mama Lou, Chris Malloy, Ambugaton, ALV, Liam Flatley, and Scooby Clark.